Hello, good evening, and welcome to all of you. This is Rajesh Kumar Singhal, and I am here to present a new lecture on the compatible solutes and osmotic adjustment. So, friend, this is also a very important topic, and in the last few lectures, we have discussed about the abiotic stresses. Okay, so in the first lecture, we have discussed the basics of the abiotic stresses. That there are different types of abiotic stress such as heat, drought, salinity, and flooding stress. And then we have discussed the different effects of the stresses on the plant growth and development. Later on, we discuss about the drought, that what is drought stress and the types of drought stress, that how the water deficit condition are generated in the soil condition or field conditions. Then we have discussed in the last class we have discussed about the drought resistance mechanism. So in this lecture we had discussed that there are three different types of plant. First one is the drought escapers, which complete the life cycle before the onset of drought stress. Second types are the drought avoidance types of plant, which generally maintain high water potential and high root rates, so they maintain high water and reduce the effect of water stress. And third types are the drought tolerance. So these are the actual drought resistance or drought tolerant plants which are able to maintain optimum physiological, biochemical and molecular processes under drought conditions. So in this we had discussed that the accumulation of these compatible solutes are the one of the mechanism by which uh, the plant can generate the tolerance capacity. So in this lecture we will discuss about the compatible solutes and osmotic adjustment. So these compatible solutes are the low molecular weight. So these are the low molecular weight compounds, and these are the highly soluble compounds, and also they are non-toxic at higher concentration, means they are not interfere with the other cytoplasmic reactions. So these three are the important features of the compatible solutes. Means which are the these three features? These are the low molecular weight compounds and highly soluble in the water, and they are non-toxic even at higher concentration. So these compound known as compatible solutes. So what is their functions? So the main function of these osmotic or compatible solutes are the maintain water relation under the osmotic stress. That's why they are known as, this is known as osmotic adjustment, because they maintain the osmotic stress. Okay, so there are different types of compatible solutes. Which were this? Also we had discussed in the last class that soluble sugars, sugar alcohols, glycerols, proline, glycine, butane. So this is glycine, butane. Okay. There are some organic acids and some ions like calcium, potassium, chlorine, and free amino acids. So these are the example of compatible solutes which used for the osmotic adjustment, means maintain water relation under the osmotic stress. So what is the mechanism behind the osmotic adjustments? So this is the mechanism. Okay, so you know that if there is a drought stress, then it causes the water deficit. Even under the salinity stress, there is water deficit. And under this water deficit condition, these compatible solutes are accumulates. Okay, so when there will be a unfavorable situation, there will be an accumulation of solutes, that is known as compatible solutes. And these compound has ability to lower the osmotic potential. So these compound have ability to lower the osmotic potential. So when they reduce the osmotic potential, then there will be a more attraction of water molecules in the cells. So when more water molecules comes under the cells, there will be a maintenance of turgor pressure, and it improves the organelle and cytoplasmic activity. So this is the mechanism of compatible solutes, that how they affect on the cytoplasmic activity. So under water deficit condition, they accumulates and lower the osmotic potential. Then they increase the attraction of water molecules and maintain the turgor pressure. So they improve the organelle and cytoplasmic activities. So this is about the mechanism of compatible solutes. Now we come to the different types of this compatible solutes. So first one is the glycine butane. This glycine butane, this is known as trimethyl glycine. This uh, quaternary ammonium. This is a quaternary ammonium compound. Okay, so they are 4 ammonium compounds, and this is known as trimethyl glycine. So this is a one of the important compatible solutes. So these compounds involved in the signal transduction pathway. Okay, we already discussed in the basics of abiotic stress regarding the signal transduction pathway. Also this glycine butane involved in the stabilization of membranes, because you know that under the abiotic stress, there is a production of high ROS, reactive oxygen species. They cause the lipid peroxidation and damage to the membrane structures, enzymes and protein. So this compound again stabilize this membrane, enzyme and protein structure. Okay, so this is the function of glycine butane. Okay, next come to the second important compound, that is proline. Okay, so this is also one of the important molecules which accumulates under the drought stress. Also proline is highly recommended or useful for the screening of drought tolerance. Okay, if a plant generally maintaining the high or generally generating the high proline concentration, then they show the high drought tolerance capacity. Okay, so what are the important function of the free proline? So first, the same as a glycine butane, it stabilize the macromolecules. Also it store the carbon and nitrogen, and that can be used for when this cell have deficiency of this carbon and nitrogen for relief. Okay, and third is the sink for the excess reactant. So when the excess reactant are produced under the stress condition, so it accumulates or act as a sink for the excess reactant. So this is the important function of the proline. Now come to the third compound, that is known as gamma amino butyric acids. So friend, this is a non-protein amino acids and act as a Jeter ions. Means this compound have both positive and negative ion. Okay, and this is also highly water soluble and functions as a detoxification of reactive oxygen species. So it maintains the stabilization of membrane. Also it involved during the conversion of putrescine to proline. So this is the important function of gamma aminobutyric acid, that is known as GABA. Okay. Now come to the next compatible solutes, that is known as citrulline. That is one of the amino acids and isolated from the water melon. Okay. So it's carry out the normal photosynthetic, means it's maintain the photosynthetic rate. Then second, it act as a ROS scavenger or hydroxyl radical scavenger. And third function is a protect DNA and enzyme from the ROS. So these are the important functions of citrulline. Now come to the fourth one, that is trehalose, or fifth one, that is trehalose. So it is also a very important osmolytes and generally produced under the resursion plants. The example of these resursion plants are Selaginella, Lepidophylla and Myrostomus flavifolia. So these are the two important example of resursion plants. Okay, so these plants are regenerate when they get the less water. Okay, because they have very high capacity of desiccation tolerance, and this trigger the ABI4 gene synthesis. So when there will be an increase in the ABA, so ABA is also known as stress hormone. So it provides the stress or dehydration tolerance. Okay, so this is the function of trehalose. So there are also some important enzymes like P5CS, that is pyroline 5 carboxylase dehydrogenase, reductase, carboxylate reductase, that is P5CR, pyrroline carboxylate reductase and pyrroline carboxylate, this is synthetase. Okay, so these are the two important enzymes which have important role in the proline biosynthesis. And next one is the CMO, that is choline monooxygenase. Okay, and BADH, that is known as butane aldehyde dehydrogenase. Okay, so this involved in the glycine butadiene synthesis. Then glycerol 3-phosphate dehydrogenase, it's involved in the synthesis of glycerol. So these are the important compatible solutes. So in this class we have discussed that, पहले हमने डिस्कस किया कि what are the compatible solutes, means these are the low molecular weight होता है, highly soluble होते हैं, non toxic होते हैं, means they not affect the cellular reactions. Also what are the mechanism that under the water deficit or osmotic stress they accumulates and lower the osmotic potential, which attract the more water and maintain the turgor, which improve the cellular or organelle cytoplasmic activity. Now come to the different types of compatible osmolytes like glycine butadiene, gamma aminobutyric acids, and proline is there, trehalose is there, citrulline is there. So many compounds is there. Like gamma aminobutyric, it acts as a deuterion, or also it is highly water soluble. Okay, glycine butadiene, it involved in the signal transduction pathway. Then free proline, it acts as a sink for the excess reductant. Trehalose, it involved in the dehydration tolerance or desiccation tolerance. Citrulline, it carry over the normal photosynthesis, also protect DNA. So means you can say that there are so many types of compatible solutes which mainly involve in the detoxification of ROS, stabilization of membrane enzyme proteins, also and maintaining the water relation under the stress condition. So this is about the compatible solutes. Hope you understand the today's lecture regarding this. Thank you very much.